Good morning, ladies and gentlemen. It is my great pleasure to introduce to you today Dr. Jaime García-Añoveros, a renowned researcher in auditory neuroscience. Dr. García-Añoveros has an impressive academic background, receiving his PhD from Columbia University. He then served as a postdoctoral research fellow at Harvard Medical School and Massachusetts General Hospital. He has since held several faculty positions at Northwestern University, Chicago, making significant contributions in investigating the molecular and cellular mechanisms underlying sensory perception. He is also a dedicated educator and mentor, inspiring many students and researchers who have gone on to make significant contributions of their own. Dr. García-Añoveros has received numerous research awards and honors for his research, and today we honor him with the 2023 Bellucci Pioneer Award in Hearing Research. His presentation today is A Developmental Dichotomy, How the Cochlea Makes Inner versus Outer Hair Cells. We are honored to have him here with us today, and we look forward to learning more about his research and insights. Please join me in welcoming Dr. Jaime García-Añoveros.
Did you take? Let's have one. Oh. Okay, thank you. So these are the outer hair cells and the inner hair cells labeled with their more characteristic proteins. The prestin, it's the protein that makes outer hair cells electromotile. Vesicular glutamate transporter is what really makes, is part of what makes inner hair cells very strong presynaptic cells. So just as a background, these two hair cells are aligned along the cochlea. The inner hair cells are the bona fide sensory receptors in that they signal to the majority of the neurons, and that's pretty much how information goes to your brain from sound. The outer hair cells have this unique feature of being electromotile. So when they get stimulated by vibrations, they contract and expand and further amplify. That's how we can hear very soft sounds and also distinguish very similar frequencies. They're a unique acquisition of mammals. I am going to, obviously, in attempting to regenerate hair cells, it's very important, as you will see today, and you probably know, you need to put inner hair cells in the position of the inner hair cells and outer hair cells in the position of the outer hair cells. If you don't do that, you actually interfere with hearing, okay? An outer hair cell here or inner hair cell here messes up hearing. So what we come in is trying to figure out how the inner and outer hair cells are differentially made. And our story developed from, Okay. A story developed, goes back to when I was a graduate student with Ann Duggan. And we were both graduate students in the same lab. And we were working in C. elegans, and I worked on the touch neurons and the functional genes, and Anne worked on their development, and she identified a gene, AGO46. There are two types of mechanoreceptors there, harsh touch and light touch. And in this mutant of AGO46, the harsh touch neurons became light touch neurons. Now, we set up a lab years later and decided I'm going to see what the mammalian homologs do while I was working on ion channels. Since then, the whole lab is doing what Anne brought in. What happened is we found out that the ortholog of AGO46, one of the two, is INSM1. And after several years working on it, we looked later and we found that it's in one type of mechanoreceptor and not the other, the outer versus inner hair cells. So we figure, okay, what could it be doing something like what it's doing in C. elegans, even though the cells are not homologs? And we ablated INSM1 conditionally. The complete deletion is embryonic lethal, although we can culture the organ and see the same phenotype. But essentially, when in all my figures, I'm going to show the control on the top and the conditional knockout at the bottom. In this particular case, I use an HO1 Cree INSM1 flux that gets rid of INSM1 in all the hair cells. And as you can see with different markers, we find out that many of the cells in the position of the outer hair cells display characteristics of the inner hair cells, where there is loss of resting and acquisition of big GLUT3, loss of calmodulin, acquisition of oncomodulin. The stereocilia shown here in white are characteristic of the inners, even though those cells are on the position of the outers. The shape of the cells, the size of the nuclei, the position of the nuclei, pretty much anything we looked at, the fact that they have a lot of presynaptic structures, the inner hair cells with stop little dots, and that nuclear CTBP2. It's an antibody that labels two things, the synapses and a nuclear protein. And the transformed cells have more synapses, as if they were inner hair cells. Electrophysiologically, the transformed cells, which we refer to as outer compartment inner hair cells, because they're inner hair cells in the position of the outers. They lose expression of a potassium channel that is typical of the outers and acquire one that is typical of inners, and that's reflected on their electrophysiological properties. More importantly, and not surprisingly, since they lose prestin, they lose electromotility. Okay, so just going very briefly, it's not like the outer hair cells, the inner hair cells move into the position of the outers, because you have the same number of inner hair cells and the same total number of cells in the outer compartment. Just half of them now look like inner hair cells. It's also not that they died and you're getting regeneration from supporting cells, which happens postnatally. We ruled that out. What we were thinking is either that inner hair cells are born in the position of the outers, or outer hair cells switch their phenotype, the transdifferentiate. And it's something similar to what Jenny Stone just mentioned for the vestibular system. It's the second that we really find happens. So the first thing is we are having an INSM1, maybe I could go back, an INSM1, these are alleles that we're going to talk about. The wild type allele of INSM1, one single exon, a knockout allele that has, it's completely deleted, or another knockout allele that replaces the coding sequence with a GFP, but also with the Cree recombinase, okay? So we have these allele over these, we have effectively a heterozygote. But you can see INSM1 gene expression in green. If we have this allele over this one, we have no INSM1 protein produced as a mutant, but you also see INSM1 gene activity. And you can see this is the control, and these are animals that basically don't express ever any INSM1 protein, but they express INSM1 gene in the outer hair cells. So the outer hair cells think they're outer hair cells when they're born. Perhaps more convincing, what I used is later on in this study, we found out other markers of outer versus inner hair cells. And one of them is transcription factor BCL11B. Now, again, the control is on the top, and you can see that early on, it's in the outer hair cells and it stays in the outer hair cells, okay? In the absence of INSM1, initially it's in all of the outer hair cells, but then it gets, within a couple of days, starts going down in many of them. These are now cross sections. FGF8 this is in situ, is in the inner hair cells throughout their development, never in the outers. In the mutants, it's originally not in the outer hair cells, but within a day or two, it starts being expressed. So by birth, we have a complete switch of about half of the outer hair cells into inner hair cells. There is, I think we can figure out that INSM1 does not initially establish the outer hair cell fate, but acts later to consolidate it. INSM1 has a expression of throughout the embryogenesis and early postnatal stages, but we also know it has a window of action that is very limited. So in the previous case, I was deleting INSM1 with a promoter that acts before its expression. But if we delete it with its own promoter, So INSM1 Cre deleting INSM1 flux, or with GFI1 Cre, which is at the same time. There we have perfectly normal ABRs, no defects in hearing at all. And what we get is only about 0.5% of the cells transdifferentiating. So INSM1 is acting during a very brief period of time to consolidate the fate of outer hair cells and preventing them from becoming inner hair cells. Okay. So in order to figure out what it does, I'm going to try to go quickly through these slides. We first sorted outer hair cells with and without INSM1, as I mentioned before. INSM1 GFP over a wild type, or INSM1 GFP over a deletion. And find out, this is the whole transcriptome, genes that were potentially up or down regulated by INSM1. Okay. It didn't really tell us much. Now, bear in mind that only half of the cells transdifferentiate, so I think a lot of this is probably noisy. So what we did is, okay, we wanted to know, is INSM1 activating outer hair cell genes? Is it inhibiting inner hair cell genes, or is it doing something else? So when we first did this way back then, we didn't have a transcriptomes of inner and outer hair cells, but this gave us this allele that we have of INSM1. I don't know why this is not. Okay. Gave us the possibility of labeling the cells. It's very simply explained. The INSM1 GFP Cre can delete TD tomato, and we can also have green expressed through other factors. So maybe I'll explain it here. Oops. We basically had an easy strategy for labeling all the hair cells in green with ATO GFP and only the outer hair cells in red with the INSM1 Cre. And we could fact sort thousands of them and do a normal RNA-seq, bulk RNA-seq. And we essentially found out these are all the differentially expressed genes. We tested everything with a blue arrow was already known to be differentially expressed. Everything with a black dot, we tried to confirm, we retested RT-qPCR. So here we're comparing RT-qPCR with RNA-seq values. And this was important because genes like BCL11B or MSX1, which are expressed at low level in one cell and not at all in the other, we wonder if they were real, and they are. Genes that were only twice as much in one cell versus the other, we thought they could be noisy, but they are real. By the way, I think this should be more prominently portrayed in the DEAR database, because I swear this is, I think, the best database for finding out if something is in inners and outers at first. So I'm publicizing it. We confirmed these genes by in situs, reporters, everything. So maybe one little thing is that when we compare our transcriptome with the one that David Hay had found in adult outer and inner hair cells, we find is very little overlap. And I think this tells us something about the transcriptome during development is very different. Now, this is a time where the bundles are being produced, a lot of cell differences are being formed during development, but not later on. And so it's kind of important to know. But going back to what we wanted to do, I'm going to jump through this because it's just, so the idea is INSM1 activating inner hair cell genes or repressing outer hair cell genes? Is it activating outer hair cell genes or repressing inner hair cell genes? It's activating, it's preventing inner hair cell gene activation. And in reality, when we tested all of these candidate genes by RT-qPCR, we found out that most of them were false positives and only the ones in this overlap. So inner hair cell genes and rich genes actually went up in INSM1 mutant outer hair cells. Okay, so what I plotted here is only some of the genes of the many that are not differentially expressed early on. These are genes that are, this is inner versus outer hair cells. So genes that are preferentially expressed in the outers are here, like Prestic. And here there are genes that are preferentially expressed in the inner hair cells, like BGLU3 or otoferlin. And then the few genes that we thought were upregulated in the mutant versus control outer hair cells, the ones that lack INSM1, which convert into inner hair cells. And what it is most of those genes are normally expressed in the inner. So what INSM1 is doing is initially preventing the expression of a core set of inner HER2 specific genes. Okay, and we confirmed that by in situ hybridization. And when you do in situs, we were expecting two patterns. I'm not sure we get the two patterns, but I'm showing them here with preliminary data. So if INSM1 is required for expression of the genes, since we delete it from all the outer hair cells, it should go up in all the outer hair cells. On the other hand, if the gene is a gene expressed in the formation of the inner hair cells, only the ones that are becoming inner hair cells would express it. And the majority of the genes that we find are in that category. So I'm going to detour here for a moment. You've seen throughout that only about half of the outer hair cells become inner hair cells. And this is not because of an impartial deletion. Even with a complete deletion of INSM1, which we can culture, we get about half of the cells transdifferentiating. And what we notice is that the cells closer to the inners are more likely to transdifferentiate than those farther away. And that really what suggests or what reveals is really that there is a gradient and suggests that INSM1 renders outer hair cells responsive to a lack of INSM1, renders responsive to an INSM-inducing morphogen, inner hair cell inducing morphogen. And as evidence that this is indeed the case, this is our sort of schematic of what we think it happens. INSM1 is expressed early on. It represses expression maybe of some inner hair cell, early inner hair cell specific genes. There is a morphogen, and INSM1 prevents a response to this morphogen. But if you don't have INSM1, the cells can express the morphogen, and the ones closer to the inner hair cells are more likely to become inner hair cells. So as evidence that this is happening, and I'm not really ready to discuss the specifics today. This is work from Gemma, and this is going to be her next big paper, hopefully the one that'll give her a faculty position. But basically, you have here the mutant, okay, and you have a mixture of inner and outer hair cells. And with activators or inhibitors, we can drive to make all of the cells transdifferentiate or very few of them to transdifferentiate, okay? So we have evidence of a morphogen, and of course, Matt Kelly and many other people several other people have been making a lot of progress into finding out different morphogens acting. We're looking for the one specifically inducing this effect. That is the control, okay. So now I'm gonna mention something else, work in progress, that we're doing with the help of Litau, which is how do we think INSM1 is really working, okay? And one of the things that we did is, it's preliminary data, so I won't really be able to answer that, but I'll tell you a little bit of what we did. We separated inner and outer hair cells. This is so annoying. And this time, instead of doing RNA-seq, we did ATAC-seq, okay? And we find open chromatin. These are two genes expressed in both inner and outer hair cells, and we can see the promoter and perhaps an enhancer or a part of the promoter open, equally accessible in two populations of outers and two populations of inner hair cells. But then when we look at genes that are expressed typically only in the outer hair cells, like NeuroD6, this is the direction of the gene, or INSM1, we see the promoter only accessible in those cells and not in inners. On the other hand, if we look at the genes, a couple of genes that we find are specific to the inner hair cells expressed, we find the promoters and perhaps enhancers only accessible in the inners and not the outers. Our working hypothesis is that maybe INSM1 is acting as a chromatin modifier during this transient period, so that the morphogen cannot activate, let's say, TBX2 in the outer hair cells because it's shut down. But it's just a hypothesis we're working on. So this is now our current model of how INSM1 may be acting. It really maybe modifies the chromatin. It prevents response to a morphogen. And if on the other hand, you don't have INSM1, the morphogen can activate certain genes and turn them into inner hair cells. So from these genes, these early genes, we thought, well, maybe we can find here what's essential for making an inner hair cell, because when these genes are expressed, the cells become inner hair cells. So going back to the data set that we had about 20 genes to look for. And again, put the spot on the right one, TBX2. Different people work in different ones, but she thought TBX2 was the most promising. I didn't. I was skeptical because TBX2, if you look at its in situ pattern, it's in inner Hurst cells and it's not in outers, but it's really present all around the scala media. It's missing just from the outer compartment. And it's something that's carved out as development proceeds. Early on, it's everywhere. Okay, and that's what happens. In the mutant, it goes up, however, in some outer Hurst cells. And what we did, really in response to a reviewer's comment, but it was a good comment, is to find out that whenever TBX2 was expressed, there was expression of the earliest functional marker of inner Hurst cells, which is VGLUT3. So expression of TBX2 correlated perfectly with formation of an inner Hurst cell, expression in an outer Hurst cell. Okay, so we deleted TBX2, again, conditionally. This time, again, the control is on the top and the mutant is at the bottom. And these are in situ hybridizations of embryonic hair cells. We see that markers of outer Hurst cells like INSM1 and BCL11B are now expressed in the inner Hurst cells, whereas markers of inner Hurst cells, particularly FGF8, is completely gone, and BRIP1 and MSX1, which also labels all the supporting cells. Okay, so what happens when these cells mature? This is now in the adult, this is a busy slide, and I think it's important to beat it to the ground. We're not changing one or two markers, we're completely changing the identity of the cells. So the inner Hurst cells, again, controls on the top always. The inner Hurst cells lacking TBX2 express outer Hurst cell prestin. They don't express inner Hurst cell VGLUT3. They express outer Hurst cell KCNQ4 channel, but not the BK typical of the inners. They lose expression of CALB2. They don't have the nuclear CTBP2, and they have much fewer ribbons, one or two, which is what ours normally have. The stereocilia, which is different, it's got a different arrangement in outers versus inner Hurst cells. The stereocilia, it's arranged like that of the outer Hurst cells and expresses PMCA2. And finally, perhaps more physiologically relevant, the transformed cells are electromotile, which is maybe reflective that they express prestin. So we have a complete transformation of inner Hurst cells into outer Hurst cells. The nuclear size, I'll go through this as a quantification. The nuclei is larger in inners than in outers, and the transformed cells, inner Hurst cells, basically have small nuclei, and they're positioned at the bottom. Okay. When we did an ABR, we found absolutely no response, basically no communication between hair cells and the brain that we know of. But why would that be the case? If you have outer Hurst cells in the position of the inners, if they get innervated, in theory, they could still signal. There is a feature of outer Hurst cells that I haven't mentioned here, which is that they are chemorepulsive. We and others have found out that they don't get innervated by type 1 afferents because they chemorepel them. So here with CALB2, we're labeling the inner Hurst cells and the nerves. And we're also labeling the synapses, which you hardly see with the red dots, okay? And this is the mutant. And so the inner Hurst cells that have become outers now chemorepel, so the nerve comes in and stays at about 10 or 15 microns, and it fails to contact the hair cell. So naturally, you have absolutely no communication between cochlea and brain in terms of type 1 afferents, the afferents. The distortion products are still present as expected from normal outer Hurst cells. They are reduced, and if I have time at the end, I'll tell you why. So it's an interesting after effect. But the outer Hurst cells are really not affected in this mutant. So what we have so far is in the absence of INSM1, some cells express TBX2 and become inner Hurst cells. Others, however, continue to form TBX2. In the absence of TBX2, the nascent outer Hurst cell, inner Hurst cells become outer Hurst cells. So what happens in the absence of both? I'm going to tell you that as we would have predicted, TBX2 is epistatic to INSM1, meaning all the inner hair cells convert into outer hair cells in all the with all the markers that we looked at, but none of the outer hair cells converted into inner hair cells. And that is kind of what we're thinking. INSM1 is necessary for inner hair cells to differentiate as inner hair cells and not outer hair cells, whether normal inner hair cells or transdifferentiation. TBX2 is really not essential for outer hair cell differentiation. It's important to prevent outer hair cells from responding to an inner hair cell inducing factor that activates TBX2. And that is indicated here. Now, we've seen that TBX2 is required early on for inner hair cells to become outer hair cells, to not become, to stay as inner hair cells. But unlike INSM1, which is transiently expressed, and I show you it acts through a critical window of time, TBX2 is expressed throughout development and into adulthood. So what happens is it still playing a role after the cells are being specified? And so what we did was to eliminate it postnatally with using an FGF8 tamoxifen inducible Cree that Mike Deans gave us. It's really nice because this is only expressing inner hair cells. It only works until about P9, P11, if you go more apically in the hair cells. But when you eliminate with TBX2 postnatally, at any time we've done from P2 to P9, okay, you get absolutely no ABRs, as you will see all the hair cells are converting into outers. You have perfectly normal distortion products. You're not at all affecting the outer hair cells, okay? And for anyone who's not an auditory person, I should have explained this before. Distortion product autoacoustic emissions is an unusual measure applied in the ear where it tells you the function of the outer hair cells without telling you the communication between the inners and the brain. So it's an easy way of distinguishing inner versus outer hair cell defects without even looking into the cochlea. It's one of the nice things of our system. Okay, so as we delete postnatally, we got also a nearly complete transformation. So here we're filling out the cells with TD tomato, but these ones have TBX2 and these ones don't. You can see the nucleus of the inner hair cells, which is larger in the middle, is now at the bottom and it's smaller. And the cells are cylindrical as opposed to flash shaped. Every single feature that we looked of the inner hair cells is lost, and every single feature of the outer hair cells is acquired, including expression of PMCA2, which is a marker of the stereocilia. The one exception is precisely here. It's the stereocilia of the inner hair cells. This is a control. Doesn't get rearranged postnatally into like that one typical of the outer hair cells. Now, I don't know if this is because TBX2 is losing its power, but I don't think it's that. I think what happens is that stereocilia is only made once in development earlier on. I think David Hay had a very nice paper ablating them and showing that if you eliminate them, they don't reform. They have to form at a certain time, and they form either in this V shape or in a flatter, a longer shape. They keep growing, but the arrangement is set up early in development, and I think that's an irreversible feature. I think that opens up a very interesting line of research. We have a transcriptome at this time, and I would really love to find out what determines the shape of the stereocilia. And I think we have the tools to figure that out, and it will be very important for regenerating not lost hair cells, but hair cells that have lost their bundles. But anyway, what we can conclude is that TBX2 not only specifies inner hair cells, but also maintains them throughout differentiation. So the next experiment for us is, this is now the result of our finding. If we remove TBX2, they become outer hair cells. Now, typically when you transdifferentiate, the cells de-differentiate and then redifferentiate. And I'm gonna tell you more or less quickly that that's not the case. And that is because when you de-differentiate, you would express, oh, this is bad. Let me see. Yes, when you de-differentiate, you would go back to express SOX2, which is in nascent cochlear hair cells and in supporting cells. But we never see expression of SOX2 when these cells become these cells. okay? What we did is eliminate TBX2 at P0 and look for the next week every day, and we never saw that. We also never saw upregulation of INSM1 or BCL11B. So the cells don't recapitulate, don't they differentiate, don't recapitulate out of her cell development. We think they go directly. The data's here, but I don't want to waste time through that data. What I want to tell you is, let's say 5 days after the cells initiated the transdifferentiation, you have a hybrid cell, which is still expresses the inner hair cell marker VGLU3, but it's already expressing a mature marker of outer hair cells, prestin and PMCA2. So eventually the cells keep this expression and lose that. Okay, so it's a direct transdifferentiation, which I believe is fairly uncommon in transdifferentiation in general. And it tells you the ability these cells have to transdifferentiate, at least at the postnatal stage. So our last question is, okay, so we know that if we remove TBX2 early or late, the inner hair cells become outer hair cells. But what happens if we put TBX2 in and outer hair cells? That is, it's necessary, but is it sufficient to make a hair cell into an inner hair cell? And what we did here was use organotypic cultures of the organ of Cori, so they don't look as nice, and we infect them with a virus that expresses a red protein, M-cherry, and TBX2. Okay, now this is a mutant that lacks TBX2. So the inner hair cells express the outer hair cell marker prestin. But if they have been, because they lost TBX2, but if they express TBX2 ectopically, then they don't transdifferentiate. So this is just to show that the system works. It's proof of principle. We can rescue basically the loss of TBX2. So the real experiment is here to the right. If we put now TBX2 force expression in outer hair cells, we see that they start expressing the inner hair cell marker VGLU3, and they fail to express the outer hair cell marker prestin, which is incipient now, so it's not so strong, and also the PMCA2. Those are the only two markers of outer hair cells that we could get this early on in cultures. But it's pretty telling that it seems that TBX2 is necessary and sufficient to make a cochlear hair cell, not another cell, but a cochlear hair cell, an inner hair cell, and not an outer hair cell. And this is why this is a summary of all data. This is why we say that TBX2 is a master regulator of inner versus outer hair cell differentiation and maintenance, at least maintenance throughout development. Now, this, let me see what comes next. Yeah, this is a little bit of a summary, and then I'll get into something I like to mention typically at the end of this talk. INSM1 acts briefly during an embryonic period to prevent outer hair cells from responding to an inner hair cell inducing morphogen, which triggers activation of TBX2. That's the role of INSM1. We think it acts as a chromatin modifier, but we don't know. It's a working hypothesis. TBX2, though, is the master regulator that will make a cochlear hair cell to differentiate as an inner and not an outer hair cell. Now, when we started this and we hypothesized the role of TBX2, we thought it would be necessary for inner hair cells to differentiate, but we didn't expect that they were going to become outer hair cells. Okay, the idea was that you would get an undifferentiated hair cell. And that made us thinking this is odd because outer hair cells are really the new cell type in mammals. It's only exists in mammals. And they are atypical, right? They're electromotile. They don't send much information to the brain. So why is it that they are the developmental default? And also, I think actually it tells us something about, and here I'm getting very speculative, but like anything in evolution, of how the cells, the outer hair cells may have appeared in the line leading to mammals, okay? And I like to think something like that about type 1s or type 2s, but I haven't given much thought. We should talk about it, Jenny. But effectively, what you have is, when I looked at the nuclei, one of the things we noticed is the inner hair cells have a thick, a large, big nucleus. But originally, early in development, they have a small nucleus at the bottoms, like mature outer hair cells. In the inner hair cells, several days after birth, first it blows up, and then it goes to the middle, okay? Okay, this is evolutionary time, and at a very different time scale development, okay? So what we think is, ancestrally, in an ancestor to mammals, you would only have cells like inner hair cells. The inner hair cells are the typical hair cells in that they have a prominent presynaptic apparatus. They're densely innervated. They send a lot of information to the brain. Okay. All it would take is at some point, loss of TBX2 in the more distally located, the more lateral located hair cells. Maybe because they express INSM1, or maybe because morphogen got reduced in quantity or in effectiveness. But now what you have is an undifferentiated cell with a nucleus at the base who basically doesn't get properly innervated. And it will be a useless cell, except what you then need is another sort of loss and gain of function mutation. You have a transporter, Prestin. Prestin is an ion transporter in non-mammals. But in mammals, it mutated, it stops from being able to transport, and instead it moves when it tries to transport, and it's become electromotile. So now the cells have acquired a new feature. So by basically having two failures, you create a new cell type, and now the cells can amplify sound. It's just a thought, but I like it. So it seems that do I have time? If I'm running out of time, I can finish here. I'll just, I can answer questions. I do want to thank people. And I have been working on this forever, right? She kind of led this project. Kazu does the single cell physiology, Marianne Cheatham. These are my two great collaborators at Northwestern. That's all the hearing tests with Yingye. And then, Gemma is looking for jobs, and she's doing the gradients, and she's done great innovation projects. Chuanzi did the transcriptomics with the help of Litau, Neil Siegel, originally in Litau now. I think that's, it's listed in many of the papers. They did a lot of the INSM1 work with John Clancy. Thank you very much.
Any questions from the audience?
I mean, this is always really great stuff. It's really interesting. When you delete TBX2 later, so like P9, what happens to innervation of the inner hair cells?
That's coming from you, and not surprised, a good question, but that's something that one of the things that I can't really, that I didn't mention here, but we're working on. We have a paper in progress. So if you delete it embryonically, the nerve touches and comes back, because the repulsion is based on contact, is based on ephrine and semaphorin, okay? And they stay like this. If you delete it at early postnatally, it stays in contact. It doesn't repel anymore, but it doesn't form synapses. The ABR is still gone. If we do it at T11, which is only in the apical hair cells, synapses are formed and they will stay. So the repulsion is limited, and once the synapses are made between the hair cells, they will stay. So the cell changes its features, except now at P11 they still keep the contact, and then we can get an ABR, which we didn't before. So it's one of the things. So a follow-up paper we want to work on, because we can learn a lot about the innervation and the critical periods by switching the cell.
Very nice talk. Here, David. Yeah. So when I looked at our transcriptome data from zebrafish, the zebrafish hair cells do express TBX2. So that's probably would be, a very interesting model to test whether if you knock out not Prestin, TBX2, whether that will convert maybe type 2 hair cells, or maybe those type 2 hair cells have some kind of a motility or something like that.
Well, okay, zebrafish, they don't have two types of, they don't have a cochlea. So they don't have outer hair cells, and their Prestin is not electromotile, right?
Right.
So, but the idea is that you might get an undifferentiated hair cell, maybe one, I don't know if the nucleus moves up. One of the things that really puzzled me is I always thought it was gonna be the other way around, right? The outer hair cells should bring down the nucleus. But actually, that's how they begin. And the outer hair cells need to have the nucleus down because they're contracting and expanding. So if in the mutants, when it goes up, they damage it, right? And you get a hair cell loss. It's the other way around, but that would be an interesting experiment. Yeah. And in chicken, I think someone asked last time I presented, I haven't looked in chicken. I don't know if Nesfrain, do you know if TBX2 is expressed in chicken hair cells? All of them? But they don't know. We should look. I mean, the data's there. I should have looked. I just haven't.
That was really a wonderful talk. I just, it looks like TBX2 is such a central factor here. I mean, is there anything known about target genes, or how are you thinking about addressing that?
Yeah, so we started doing transcriptomics, right? And I figure I'm gonna do it bulk. And we add tamoxifen at P0. And we collected at different time points, but the earliest we can collect is 1 day later, okay? 1 day later, which is amazing. There's already signs of transformation and hundreds of genes are already being misexpressed. So I was hoping to look at the very first few factors, but I don't have a resolution because I need that cell to become red. So I have to figure out another way of doing it. Because too many things have changed, but we already have expression of outer hair cell genes in the inner hair cells within 24 hours. It's amazing because TBX2 is a very highly expressed mRNA and the protein, and you think in 24 hours, I thought there would still be plenty of it left, but it acts very quickly. It must have a very short lifespan, half-life.
Thank you.
Okay. Thank you.
So